My name is Siyuan. I am a graduate student and new to medicine. Today I will talk about genomic diversity and invasion history of Drosophila suzukii. So, Drosophila suzukii is a highly invasive crop pest and currently a major pest species in America and Europe. It has rapidly invaded the whole globe during the past few decades, occupying a range of distinct environments. The adult female flies lay eggs into unripe summer fruits with their ovipositor, and the infection costs $1 billion of crop loss per year. Current knowledge about the invasion history comes from individual reports and genetic inference of invasion routes. The genetic inference was based on limited microsatellite markers, which argues for an Asian ancestry. With the species, we're curious about how much diversity is in different natural populations and what is the invasion history reflected from its genome. To study those populations, we collected fly samples from Eastern and Southeastern Asia, North and South America, and Europe. With a total of 29 population samples from our own collection and another research group, we built a global set of whole genome pool sequencing data. After variance calling with our customized pipeline, we studied within population variations as well as population differentiation and migration using summary statistics, principal component analysis, and tree construction. We first compared the genetic diversity of each population. The clear drop in nucleotide diversity of invasive populations relative to observed diversity in the native range, including China and Japan, suggests strong founder event bottlenecks. The X chromosomes have lower diversity than autosomes, with effectively longer bottlenecks due to their lower effective population size. The population with the highest diversity is the East China population. We inferred that all other populations have a subset of the genetic diversity present in Eastern China, consistent with the ancestral or refugial species range in this region of Asia. Another piece of evidence that also supports invasive bottlenecks is that invasive populations have lower proportion of rare alleles than native ones, which are easier to lose during bottlenecks. We then looked at population differentiation using principal component analysis of allele frequency. From the PCE plot, we saw clustering of populations by invasive ranges, then further grouped into continents and islands. An interesting pattern is the deviation of three populations from the United States mainland. The decreasing distance from Hawaii to Oregon, then to Middle California, suggests the invasion route from Hawaii to the west coast of US. The heat map matrix of genetic differentiation recapitulates the clustering of distinct native and invasive populations. The additional information is that two island populations of Hawaii and France Réunion are more differentiated with all other populations, which suggests a more isolated population history. Next, a hierarchical population structure predicted by neighbor joining tree of allele frequency suggested separate Asia-sourced invasion events into America and Europe. The West Coast invasion pattern from Hawaii is also reproduced here. And last, a maximum likelihood tree supported the inference that all other populations are found to have a subset of genetic diversity in Eastern China, The tree-based mixture inference predicted gene flow among Europe and America following the first founder events. So in conclusion, we found that the highest genetic diversity is in East China, which could indicate ancestry in Southeast or Eastern Asia. We also found that strong founder bottlenecks during early invasion events. We also conclude that Asia-sourced invasions into America and Europe are independent from each other. After figuring out the genomic diversity and invasion history, we're then interested in how those populations adapt to distinct environment. The first part of our ongoing work is to search for target loci of local adaptation using environmental association analysis. We're separately looking for loci that are associated with 9 uncorrelated environmental variables, representing variations in climate, geography, and habitat. And the next step is to identify inversions and copy number variations that are derived by invasions. Thank you.